Hey everyone, welcome to the Chapter 2 Chemistry of Life Notes. This is Part 2, Carbohydrates. Before we start talking about the macromolecules, we're going to talk about functional groups. Functional groups are small groups of atoms with specific chemical properties. Functional groups confer these properties to larger molecules. For example, a molecule can be non-polar but may have a functional group that is polar, so that one section of a larger molecule would be considered polar, just that one section. One biological molecule may contain many functional groups. And so there are several functional groups we're going to see in this course. For example, the hydroxyl group, which we saw in the properties of water. So the hydroxyl group is an oxygen and a hydrogen. Remember that oxygen is more electronegative and will take the electrons away from that hydrogen. And so this is a polar functional group. This dash right here would be the bond where the oxygen connects to the rest of the larger molecule, whatever it is. Next is the carboxyl group, which is COOH. Now, written like this is the simplified form. There may actually be the carbon bonded to this oxygen and then the hydroxyl group coming off over here. So you may see this in a slightly different form depending on what we're looking at. For example, an amino acid versus some other molecule, but this is just the simple way of writing it. Next is the phosphate group, and the phosphate group is also has a lot of side atoms to it, so we're going to draw the simplified form for this class. And so this is what the simplified forms looks like. I tend to circle the phosphate group just to remind you that it is a group. There's other items there, but we're not showing them in our simplified version. Last is the amine group, and it looks like this. Again, this is a simplified version. You actually may see different branching off of the nitrogen, but we're just going to keep it simple like this. So these are the functional groups you're going to need to know for our course. So for the next part of Unit 2, we're going to be talking about the macromolecules. Macromolecules are larger molecules, and most biological molecules, so many times students remember the term biomolecule from 9th grade, are polymers. Now, polymers is a word that is two roots combined. Poly means many, and mer means unit. So a polymer is literally many units. And they're usually held together by covalent bonds of the smaller molecule called monomers. So you can think of this like the links in a chain, where a single link would be called a monomer, but when we put several of them together, it is referred to as a polymer. When we look at organisms, living organisms are mostly made of water, as you can see from this pie chart. There are other ions and small molecules, but macromolecules make up a chunk of what living organisms are made of. And of these macromolecules, there are 4 types. We have proteins, nucleic acids, carbohydrates, and lipids. And as you can see from this portion, as we break down the percentages, what would this be made of? The majority of the macromolecules are going to be proteins, followed by nucleic acids, followed by carbohydrates, usually in the form of polysaccharides. And then the smallest portion is lipids. Polymers are formed and broken apart in reactions involving water. There are 2 important terms you need to know for the formation and the breakdown of these polymers. The first is called condensation, which is the removal of water, and that's going to link monomers together. In other words, condensation reactions build up the polymers, and sometimes you'll see them denoted, or the process of condensation called dehydration synthesis. Hydrolysis is the addition of water, and this process is going to break the polymers into monomers. So these two are the opposite of each other. Your condensation or dehydration synthesis is going to build polymers, whereas your hydrolysis is going to break polymers. We're going to look at specific examples of how these processes occur in each of the macromolecules by the end of Unit 1. So let's start with the first macromolecule, which are the carbohydrates. We're going to review what you had learned from the macromolecule study set. To do that, what we're going to do is highlight from each row the characteristic that pertains or that applies to carbohydrates. So for example, in this first row right here, go ahead and highlight or circle the chemical structure of carbohydrates. The blocks that you should have highlighted or circled was this one, because CHO in a 1-to-1 ratio is the chemical composition of carbohydrates. In the next row, we are given examples of each of the macromolecules. So in this row, highlight or circle the example of carbohydrates. The example that you should have highlighted was starch, glucose, and cellulose, because these are examples of carbohydrates. One of the ways you can identify if something is a carbohydrate is look at the ending. Many times, the ending of the word, for example, glucose, cellulose, those that end in OSE are usually carbohydrates. In the next row, you are given the types of covalent bonds that form between the monomers of the polymers. For this row, highlight the type of bond, the covalent bond that occurs for carbohydrates. The box that you should have highlighted or circled was glycosidic bonds. It is glycosidic bonds that form between the monomers of carbohydrates during dehydration synthesis, and it is the glycosidic bonds between the monomers that is broken in hydrolysis. In the next row, you are given examples of functions of each of the macromolecules. In this row, highlight or circle the function of the carbohydrates. The box that you should have circled or highlighted is quick energy and structural support. Throughout the year, we're also going to talk about the role that carbohydrates have in identification. In the last row are the monomers of the macromolecules. In this row, highlight or circle the monomers of carbohydrates. The box that you should have circled or highlighted is monosaccharides. Monosaccharides are the monomers or building blocks of carbohydrates. One of the most important concepts we're going to learn in AP biology is how structure affects function. And so if you change the structure of a molecule, you're going to affect its function within the living organism. So we're going to look at two examples of how this is true in carbohydrates. In example number 1, we are going to look at the difference between two sugars, two monosaccharides. They are the sugars that make up part of the genetic information in living organisms. Those two sugars are ribose and deoxyribose. Both of these monosaccharides are considered pentoses. They have 5 carbons. So pent, like a pentagon, has 5 sides. Pentoses are 5 carbon sugars, and their structure is very similar. Now, before you draw these two sugars, there's something I want to explain about the way that these are drawn. First of all, what you see here, and again, don't draw this until we have the two examples explained. When you have this, it is a sugar, and these carbohydrates or these monosaccharides in water will form what is called a ring. So you'll see here that's kind of a circular shape. And what we do is at each of these corners is understood to be a carbon. Now, if there's not a corner, like for example here, we just simply write the C there for the carbon. And so when we look at this molecule, when we look at this pentose, this 5 carbon sugar, The way that we do this, or the way that we identify the carbons, is starting at this up here. This is the oxygen. And from here, we are going to number the carbons going clockwise. So starting from this oxygen, we're going to go, the first corner we encounter is a carbon, and that's going to be carbon number 1. Moving on, going clockwise, the next corner we encounter is carbon number 2. Moving on, going clockwise, the next corner we encounter is carbon number 3. Moving on, the next carbon we encounter is carbon number 4. And from here, this is oxygen. We don't need to number the oxygens, we only number the carbon. So from here, the last carbon, the 5th carbon in this pentose sugar is this carbon up here. So now that I have the basic structure of this pentose, I'm going to explain what the difference is, the structural differences are between ribose and deoxyribose, and then talk about how that change in structure affects the function of this sugar in living organisms. So now go ahead and draw your ribose and deoxyribose. When you are ready, resume the video. Now what I'm going to do is I'm going to start by verbally telling you what the difference between ribose and deoxyribose are. Then I want you to pause the video there and make a prediction as to what that is going to look like. Then when you've made your prediction, continue the video to see if you were correct. So the difference between ribose and deoxyribose is the presence of a hydroxyl group at carbon number 2 in the sugar ribose. Pause now and make your prediction. So now you're going to check your prediction, and this is what it would look like. So you can see here at carbon number 2, I have drawn the line connecting to this carbon, this hydroxyl group. And that's what makes ribose structurally different from deoxyribose. Everything else is the same. The difference is at deoxyribose, there is no hydroxyl group coming off. Now, these are very simplified versions. Remember that carbon has 4 valence electrons. And so there are other groups coming off of this carbon, and this carbon, and this one, and this one, and this one. But we're not going to draw them because they just kind of clutter up the drawing, and they're not important for focusing on when we're talking about the structural differences between ribose and deoxyribose. Now that we know that the structural difference between ribose and deoxyribose is this hydroxyl group at carbon number 2 for ribose, let's talk about how that affects the way that these sugars function in living organisms. Keep in mind that this oxygen, this hydroxyl group, is polar because this oxygen is more electronegative than the hydrogen. So this section right here adds a polar portion to this molecule that is not present here. Now, when we look at RNA and DNA, which these sugars are part of, we know that there are differences that you all learned in 9th grade. So for example, DNA is the genetic information of the cell, but RNA is used only in the production of proteins. And so because of this hydroxyl group, because of the structural differences, ribose and deoxyribose in living organisms behave differently. So deoxyribose, you normally see DNA, because it's lacking that group here, is double-stranded, and it is very, very stable. RNA is found single-strandedly and is usually not stable. So this is our first example of how the structure of a carbohydrate is going to change the way it functions in living organisms. The next example of how structure affects function in carbohydrates is going to be of a particular monosaccharide called glucose. You may have heard the term glucose in your 9th grade class, and maybe even prior to that. And we know that glucose is C6H12O6. But the way that the arrangement of all the carbons, hydrogens, and oxygens in this molecule will actually affect whether or not it's glucose There's different types of glucoses, but it could also be fructose, which is fruit sugar. Fructose is also C6H12O6, but it's the spatial arrangement of these molecules that give them different properties. Because it's got 6 carbons, these molecules, glucoses and fructoses, are known as hexoses. Hex means 6. So we're going to look at 2 examples of types of glucoses. They have the exact same chemical formula, C6H12O6, but the way that these 2 are arranged is going to make a difference in the molecule. So the 2 types that we're going to talk about are alpha and beta glucose. Just like before, I'm going to give you the basic structure before going into the details about how they are different. So don't draw this just yet. So here's the basic structure of this hexose. You can see here, starting from the oxygen, I've already numbered the carbons going clockwise. So carbon number 1, 2, 3, 4. There's carbon number 5 in there, and then there's carbon number 6. So given this basic structure, I'm going to tell you the difference between alpha and beta glucose. And from this, after I've given you this, I want you to pause and make a prediction as to what this is going to look like. Ready? The difference between alpha and beta glucose is at carbon number 1. Alpha glucose has a hydroxyl group pointing downward, whereas beta glucose has the hydroxyl group at carbon number 1 pointing upward. So take a minute now to predict what that's going to look like, and I will show you the answer. The answer, or the difference, looks like this. So you can see at carbon number 1, the hydroxyl group was drawn pointing down from carbon number 1. And in beta glucose, the hydroxyl group at carbon number 1 is drawn pointing up. Now, it may not seem like this is a big deal. It's like, oh, well, I just drew it one way versus the other. But it actually really impacts the function of these 2 sugars in living organisms, especially when it comes to your ability to digest this sugar. Alpha glucose is digestible by humans and other organisms. Beta glucose is not digestible by humans or other organisms, with the exceptions of microorganisms that have the ability to break it down. I'm going to have linked in the description box below a real helpful video about the difference between digestible glucose versus non-digestible glucose, and I do highly recommend you watch it at the end of the notes. So monosaccharides are covalently bonded by condensation reactions that form glycosidic linkages. So remember, monosaccharide is your monomer. We're going to make polymers by doing the process of condensation reactions, also known as dehydration synthesis. And in that process, we are going to form a covalent bond called a glycosidic linkage. So let's take a look at what that looks like. Here we have 2 monomers of glucose. I'm going to finish drawing in the rest of the molecules. So here we have 2 hexoses, and I know they're hexoses because counting the carbons, I count 1, 2, 3, 4, 5 carbons on this one, and watch, 1, 2, 3, 4, 5, 6 carbons on this one. So that's very deceiving because the shape makes it look like it's going to be a pentose, but this is actually fructose because it's got that additional carbon up there. So I've got 2 hexoses that are going to come together and form a glycosidic linkage. Now you'll notice that some of these are highlighted in a box. I've got an oxygen, an H, and an H. So 2 H's and an O, or H2O, which is known as water. So this is called condensation or dehydration because we are going to remove water. We're going to remove a little bit from this monomer, and we're going to remove a little bit from this monomer. We're going to remove from both of these combined. That's what's in the box, and that's where this water is coming from. Notice that there is an oxygen not in the box over here. That oxygen is what's going to form the glycosidic linkage right here. And that is what is referred to as a glycosidic bond. So in this case, what happens is that is the glycosidic bond that's formed. And if you do this process in reverse, if we started with a polymer and moved backwards, then what we would see is that in order to break this bond, we're going to need to add water, because at the end, what we're going to need is to have an OH group over here and an OH group over here. Now there's already that shared oxygen. This is just going to go over to one side. We'll take one of the hydrogens, bring it over here. We're left with the hydroxyl that'll go over here. So hydrolysis and dehydration synthesis are the opposites of each other. And this can run this direction or it can go in this direction depending on the needs of the organism. In this particular example, since we have glucose, and this is alpha glucose because the hydroxyl group at number 1 is pointing down, and we have fructose, which is fruit sugar, when we combine and join them through dehydration synthesis to form this glycosidic bond, this is a special type of molecule known as a disaccharide. Di meaning two. And the name of this disaccharide is sucrose, which is just table sugar. So the type of sugar you might put on your Rice Krispies or your cornflakes. There are many examples of disaccharides we are going to look at in class. Some of them you may have already heard before. For example, lactose, which is milk sugar, is a disaccharide. Maltose, which is used to make malt sugars, is a disaccharide. So the last thing we're going to do is look at 3 examples of how changing the structure of glucose affects its function. What you're looking at here are 3 boxes, and in this first box here, you're going to notice what looks like beads on a long string. This represents the polysaccharide. And in this first box here, what you're going to see is that they are all very nicely arranged. They're very linear. They are straight, long chains that are just kind of bundled side by side. Okay, so let's take a look at the 3 molecules that you are looking at. And each of these is a representation of a polysaccharide. So it is many, many monosaccharides that have been joined together and have glycosidic bonds between the molecules. So the first molecule that we're looking at is the cellulose, which is found here. We're also going to look at this molecule, which is starch, and then this molecule, which is glycogen. We're going to look at each of these and compare and contrast. First, starting with their structure. So as I mentioned before, cellulose is very unbranched, long chains of carbohydrates. Starch is slightly branched, and glycogen is highly branched. Both cellulose and starch are found in plants. However, glycogen is found in animals. Now, because they have different structures, they also have slightly different functions in different organisms. So for cellulose, this is the carbohydrate that makes up the cell wall. And that is because this is a very tough molecule. You need a specific enzyme to break it down, and not very many organisms have this enzyme. So that makes for a very strong cell wall. For starch and glycogen, they are both storage molecules. So when we talk about quick energy, this is the type of carbohydrate that we're talking about. The difference is that in plants, it is starch and slightly branched, whereas in animals, it is glycogen and highly branched. Now, for these different molecules, for cellulose, this is monomers. The monomers of cellulose are beta glucose. Whereas in starch and glycogen, the monomers are both alpha glucoses. I have linked below in the description box 2 videos that I think will really help you understand these concepts, and I really encourage you to watch them. Thank you all so much, and I will see you next time. Bye!